why these things happen that I just showed you. So the first is this phenomena where you have bacteria in these two halves, and I realized what I should have told is that there is a half into which they aggregate, which is determined by the direction of these guys. So if I flip these guys to this side, that side, everything goes to the other half. Okay, so that's why this happens is, I mean, it's probably not surprising to you that it's something like a ratchet. Well, it is a ratchet that's happening over here, right? So what gives rise to the ratchet in this case is a difference of temperatures, right? So maybe I'll just quickly say why the Feynman ratchet fails. So for those of you who don't know, so there's a wind vane in one box, and there's a ratchet and a pawl in another box. The idea is that as the gas molecules are hitting this vane, every time it turns in one direction, it's rectified by this pawl, and if it turns in the other direction, it's held over there so it doesn't turn. And the idea is that you can get some preferred motion in one direction and not the other. Of course, this doesn't work if the temperatures are the same, because what fluctuations drive the motion of this wind vane, so the wind vane will sort of go like this, and then every time it turns like that in one direction, the ratchet turns. But then the same stochasticity plays a role on the ratchet side as well, right? So this little pawl that is holding down the ratchet also is doing that. And so even though this ratchet may accumulate some directed motion, every now and then this pawl will go off, and then the ratchet will come back, right? So you'll, in effect, not get anything. But you can get it to work if the temperature on one side is higher than the temperature on the other. So if you keep the pawl cooled, then you can actually make this happen, right? All right. So in this case, what happens is the bacteria are doing a random walk, but then every time they meet a wall, they do something interesting. Their motion is actually biased in the sense that they move along a wall. Okay? So they don't go back like that with some reduce. So the probability of them doing this is much higher, right? So that is where you're breaking symmetry already, meaning that if I have a bacterium coming towards a wall, it's almost deterministic that it goes like that. But then if it's coming in the other direction, it needs a fluctuation to kick it out. Otherwise, it'll keep going. So this is a time irreversible process, right? And so you're taking this, in effect, a difference in temperatures, and you're now creating an asymmetry in geometry, in space. So you combine the two together, which means you'll have more guys doing that and much fewer guys going in this direction, right? So in effect, you create not a Brownian ratchet like that, but a correlation ratchet over here, where there's a correlation between the spatial structure and the motion of the bacteria, right? It's also, so if this was done in time, that's what we know as a flash ratchet, where you apply a time asymmetric potential, and then you can also drive motion in one direction, right? So if you have a bunch of particles which are all diffusing, and then I apply a potential which looks like that, every time they come onto this side, you'll start collecting some, and then you can keep going. So you can drive it in a particular direction. So that's what's happening here, and of course, yeah?
I'm curious, if you say have 2/3 of the you get the concentration.
Yeah, at some point, I guess you can get, equal distribution, equal motion in both directions, and then you lose the effect, surely, yeah. So I want to point out to you the fact that these are now self-propelled, they're moving, right? And there is a hydrodynamics that's associated with this motion, right? So now we must remember that we have this E. coli, and there's an ambient fluid around. And so if this guy moves, the fluid around, because so it's moving through a viscous fluid, there's a drag that it experiences, right? So there's a drag force in that direction. The net force is 0, okay? And this creates a flow field around this object. Okay, so now this means that this can interact with walls in different ways than if this was not present, right? So it's actually the presence of this flow field that makes these bacteria align with walls. Okay, if you want to think about it, so this is like this is a force dipole, right? So you have a force like that. So you can make an analogy to electrostatics. Think of a charge dipole, and then if you have a wall, so you have a mirror charge, and so you get an attraction. It's the same thing that happens over here, except instead of a charge dipole, you have a force dipole, and you have an associated flow field, and so you get attraction to the walls. Okay, so that's why this ratchet works, because of this activity. Okay, now the turbulence is essentially an instability. So what I also told you is that for example, in the V-check model, I can get alignment of birds or bacteria or any. So in this case, it is these force dipoles. So I can treat each of these bacteria as little force dipoles. So those are aligning, all right? And now they're again sitting in an ambient fluid, which means they're driving a shear that they generate. Right? Because of this alignment, the fluid around has a certain, motion, this flow field that I talked to you about, right? So now, if a few of them misalign, this shear is only enhanced, okay? Which means that if there's a small perturbation, it only grows. And so there's so this ordered state is unstable, right? And so you will have to have these breakups, right? And then you'll start to generate these swirls in there. The fact that it actually looks like turbulence in macroscopic fluids is again in the details, and I'm not going to go into that right now, right? But then I want to point out to you that just like the Navier-Stokes equation that we wrote for passive fluids, you can write an effective description for this active fluid that is exactly like Navier-Stokes, except now you have a stress here, which is internally generated, or an active stress that's in this fluid, right? So it's this active stress that gives rise to something like this. Okay. So each of these guys, so let's say it's very big now, and these guys, some of them misalign. Now they're beating, and so they're driving the fluid around them, right? So now that creates a shear. And that shear just, makes them go further away. And so, in effect, the instability grows. Okay. Right, so yeah, so that's exactly what I just told you. This ordered state is unstable, and then you get these turbulent-like motions. Okay. And so this zero viscosity, again, is a very similar consequence. If I apply a shear, right, and I have an object over here which is itself producing a flow, right, so this object is like a bacterium, like the one I just drew over here, where the head is going like this, there's a drag force, it's a forced dipole, it's pushing fluid out, right? So this is referred to as a pusher, okay? So now, if you apply a shear on top of this, it only promotes this further, right? So you can think of it as increasing this fluid motion, right? Because of its activity. And as a result, the viscosity, apparent viscosity reduces, right? But if you instead had an organism, well, there are these organisms which do another kind of motion. So this is an algae which has a body and two flagella. What this does is it does a breaststroke, right? So this guy is pulling fluid back, and the body moves forward, right? So it's a dipole in the opposite sense. So this is called a puller, right? And what that does to the stress is that it opposes the external stress. And as a result, if you take a suspension of these organisms and look at the viscosity as a function of how many you put in, actually goes up. So if I apply a stress like so, this is going in the opposite sense, right? So it's really, increasing the effective obstruction to this flow because it's pulling it back. Whereas over here, it just is aligned in, so it's also creating a flow which goes in the same direction as the shear. So it aligns with the shear, right? So these are elongated objects, they align, and then one is creating a flow field. It's a spontaneously generated flow, which aligns again with this external flow, and one doesn't. Maybe, no?
I can ask you a bunch, but I just don't see why the...
So it's the apparent viscosity that...
It seems like both the pusher and the puller nearby them create a relative flow.
So it's the direction of the flows. So here, it brings fluid from its back to the front, the external fluid. Whereas this guy is doing the opposite. Right? One is literally, so if I'm just pushing the fluid, so the fluid goes this way, and the other case I'm pulling it so it goes the other way.
The pusher is dragging fluid along with it, while the puller is really along its side.
Exactly. And then once you have an external shear that you apply to it, the fact that they align coupled with these spontaneously generated flows gives rise to these apparent viscosities.
So both are with non-Newtonian fluids, but the algae acts like cornstarch and the bacteria act like ketchup.
Exactly. So if you were swimming in this fluid, it's not that you, so if we were if we were here, sitting in this fluid, it's not that somehow we would feel no viscosity at all, right? That's what it means. No, so I'm pulling this fluid. So there's a force which acts on the external fluid in this direction. My body moves forward, right? So it's a couple of forces, one which is pulling forward, one which is the body moving. The net thing moves, of course, because it's displacing fluid. Whereas for the bacteria, it's reversed, right? Where the flagellum is powering it, right? It's pushing fluid to the back, the body is moving forward. The flagellum is powering to the back, the body is moving forward. Right? In both cases, the object itself moves forward. But it's this sense that drives the external fluid motion that causes these things. Yeah. Also in this case. It's just so if this object were held fixed, right, and this guy is doing that, right, it'll pull the fluid, whereas this guy is held fixed, it'll push the fluid. That's it. Yeah? Okay. So now, so what I'm saying is if I apply a shear on this thing, right, if I apply a shear on this thing, the bacteria align in such a way that, you know, even after I stop applying the shear, if you will, it can keep going. Right. Exactly. And the fact that, you know, it's zero, it just means that all the dissipation that's coming because of the viscosity of water is compensated for by the activity of the bacteria. So they're pumping in energy into these scales, which allows all of these things, right? So that's what really is at the crux of this active in active matter or active fluid, is that energy is being pumped at the microscopic scale at the level of the individual, right? And that gives rise to all of these interesting physics. Okay. So I must also say that this is at very low shear rates. So if you're actually able to swim with such low shear rates, you will probably feel this thing. Yeah. So that's one thing I should have mentioned. Right. I don't know. That's a very interesting possibility, but yeah, I don't know. What I thought of myself is that, you know, for instance, if I have something like this, and so I tried to think of something to run on the bacteria, and if I had a source of food on the other side, So the physics governs things to come to one side, whereas biology wants it to go the other way, and if it will actually evolve a mechanism to beat this. I don't know the answer to that, and yeah. So, right. So I'm going to touch upon evolution on a very fast time scale in a moment now, yeah. So if indeed such physical mechanisms, organisms can take advantage of, presumably bacteria can fight back in some way, and I'm going to touch upon that with antibiotic resistance and evolution in just a moment. Yeah.
Do the pushers and pullers go to the same side of this asymmetric barrier?
Pushers and pullers do, yes. Yes, so their alignment, so the asymmetry, what really breaks symmetry is the fact that they go along the wall, right? So as long as you have the same orientation of the wall, you should get the same behavior, and you'll get alignment of the wall in both cases. Okay, so now I'm going to change tack a little bit and tell about evolution, like I said. And I'm going to start with this very famous experiment that set a lot of our understanding of how mutations arise and so on, the so-called fluctuation test that Luria and Delbruck performed. So the question is this, right? So do mutations arise in response to a stress that you apply, or are they random and they pre-exist, right? This is the question. Or are they spontaneous, random? And so what they did to check this was a very, very simple experiment. So if you start with a mother culture of bacteria, right, and or you take some bacteria and put it on a plate which is infected by virus, a lambda bacteriophage, this virus kills the bacteria, right? So if, however, there's a resistant strain, it can survive and grow on this plate. Okay? So if the mutation is induced, if you take a mother culture and let it grow into daughter cultures and take those daughter cultures and plate it onto this bacteriophage, you should expect the same number of colonies roughly to survive, right? Because they're all reacting to the same stress that is applied, and you would expect that the same number of resistant mutants arise and so on. However, if it was spontaneous, then you end up with a situation where a resistant strain arose in the very first generation, and so you'll have a lot more resistant strains in the subsequent generations than if a resistant strain didn't arise, right? So if you look at the statistics of the colonies that grow on these plates, you will expect to see these plates where you have a lot of colonies growing, or jackpots, and some where you have nothing growing, right? And so just by looking at the statistics of these number of colonies, they inferred that there's spontaneous mutation in bacteria. Okay? No. So the point is that if it is induced, this is what you expect. If it is spontaneous, this is what you expect. And when they set up the experiment, this is what they saw. Okay? Induced means that, so I'm doing nothing until I face some stress, and then in response to the stress, I react, right? And then I come up with some solution to that. Whereas this is saying that, every time I divide, I make a mistake in copying, and so some of them are beneficial, some not, and some are kept, and so that gives me this result, right? And this is what they found, right? Right. So what we are doing is taking one of these things and just growing it here, right? So if something survives, there's no further. So you're just looking at a colony that's growing. You're not looking at anything subsequent to that. Yes, sure. So for instance, over here, all of these guys are resistant, right? And so all those 4 bacteria will form 4 colonies, right? And after that, I'm not looking at any further branching over there. That's where it ends. So it's almost like I'm taking a time snapshot of the state over there by putting this condition of death. Okay. All right. Yes. Maybe 50 die, exactly. So maybe 98 die, 2 will survive, right? And so those 2 will spawn 2 colonies, and that's what you can actually see, right? So they're not counting individual bacteria, but they're counting the number of colonies, meaning there was one bacteria which was resistant, and then it grew into a colony, right? You just run it once. Exactly. It's a one-shot experiment. Exactly. It's a one-shot. So that's why you're taking a snapshot over there. Right? So if there was nothing resistant, all of them die over there. And then you have no scope for, further. So they don't wait long enough that they get so many more generations. They're just looking at it, after they've grown or they've divided sufficiently long that you can actually see and count a colony. Red indicates that they're resistant. Yes.
So on the left side, none of them had developed response.
Exactly. That's the idea. So here there's no pre-existing resistance, but as soon as you put them down there, some of them are able to fight this and then get this. So this experiment basically argued against this possibility. Yes, stress is just at the end. Stress is just at the end, exactly. Yes. You get a very high, exactly. It's the distribution that gives you information about, right. In the experiment, this is what they observed, right? And the conclusion is that the mutations are spontaneous, meaning that, you're making copying errors, and then some are beneficial and some are kept, some are not, and so on. Right? So that's the idea of this random mutations that give rise. So what this tells you is that evolution, if this is the case, is a slow process. Right? So you need many generations before which you can hit something like that and then change. Whereas over here, it can potentially be fast. Okay? And I'm going to tell you about an experiment where this is actually shown to be true. Right? And you need a particular way of doing the experiment in order to get that. Okay, and this is the particular way, right? So Sewell Wright, he introduced this idea of fitness landscapes where organisms and populations are traversing this rather complicated landscape, and they're going into regions which are more favorable than others, right? And this whole journey, if you believe the previous thing, is driven by this random mutation and generation of mutants. And what he said was, if instead of having one big population that's moving on this landscape, if you broke up the population into many tiny ones and created a meta population and allowed for dispersal between these populations, then you get something very different. Okay? He said that there is now a local competition, right? And then if you win locally and you disperse to another population, you have a better chance of survival over there, right? And so, in principle, a beneficial mutation that arises in a subpopulation can spread faster through this thing. Okay? And so this idea was implemented in a very, very clever and beautiful experiment by Bob Austin and colleagues at Princeton. So what they did is this, right? So what you're seeing here is the schematic of a so-called microfluidic device. Okay? What that is okay, let me explain to you what you're seeing here. So you're seeing here many connected hexagons. Okay, so now this is a little chamber, very similar to the chamber that you saw before with the walls and so on, where the bacteria were. And there are channels which connect different hexagons. Okay? So now each little thing is a subpopulation, and bacteria sitting in one of these can migrate to another through these channels. Right? So this is a metapopulation on a chip. So this is a small device, right? And what they then do is, so if you have some hexagons at the edges, they create very, very, very fine channels, which are on the scale of a few hundred nanometer, such that the bacteria can't swim out. But if you flow some food through here, it can diffuse. All right? So that's how they provide food. So LB is the Luria broth, the same Luria. That is food for the bacteria that flows in this direction. And then in the opposite direction, you flow the same food together with an antibiotic. Okay? Ciprofloxacin is an antibiotic that kills the bacteria. Okay? So now you can visualize, if I pump in food from here, this is the nature of the distribution of the food because of the diffusion through these channels and this network, right? So you get a lot of food here, and you just decrease as you go away from it. Whereas in the opposite direction, you're driving antibiotic. So you're creating a gradient of both antibiotic and food. Okay? Sorry. Yeah, okay. Yeah. So this is yeah. So this is antibiotic plus food, and that is the food coming from the top. Yeah. So there's a gradient. So there's more food here, less food here, more antibiotic here, less antibiotic.
It also has food, right? Antibiotic also has food.
Yes.
So looking at that food is not consumed, right?
Okay, so let's make it even simpler. Let's even ignore this food for the time being, right? So it all works. So let's just say this is just antibiotic. Okay, so there's antibiotic, there's food, right? And then, so yeah, you have a gradient of antibiotic, you have a gradient of food. So what you have really is a gradient of stress. Okay, you're subjecting a population, metapopulation to the gradient of stress. So that's how this device looks. It's etched out in silicon, and you put the bacteria in there. And so there's this very special point at the meeting place of both the antibiotic and the food where the gradient is the highest, right? So then the idea is that in this very, very high gradient of stress, you can actually drive up or you increase the chance of getting a beneficial or a resistant strain, and that can migrate and populate and create a resistant bacteria in there. Okay. So you have bacteria which are swimming, right? So they can swim from one to the other through the channels.
So the key idea here of stool rate is it's not just that, it's not just that each bacterium has a small population with which it can compete. It's that different kind of populations are exposed to different amounts.
So there's a gradient or a different, yes, exactly. That's the key idea. Yeah. Same kind of stress, but it's a different amount. Yeah, it's a different amount. No. No.
So is the point of this experiment to tell whether the bacteria are able to evolve more quickly than they would if this was a
Exactly.
homogeneous?
Exactly. Exactly. So if I perform this experiment on a petri dish, like Luria-Delbruck, with just the antibiotic there, most likely I'll kill all the bacteria.
Even if it was diffusing it from the side?
Even if it was diffusing.
So there was still a spatial gradient.
Exactly. Even if it was, exactly. Okay. Okay, so now this is a movie of the experiment. Now this time is minutes and hours. So I must also tell you that whenever you see something green, it means it's resistant to the antibiotic and the bacteria is actually growing, right? If you don't see green, it means that the bacteria is just dying. So the bacteria is expressing a GFP, right? So if you see nothing, as here, there's no bacteria. So you, all right, so I must also say one more thing, that you initially inoculate the seed over here, right in the center, and then the bacteria go out. Okay? So there you put some bacteria in, very quickly die out. Seemingly nothing happens for a very long time. And that's the very special Goldilocks point. Already at around 4 hours, you see bacteria which are growing. And then very soon, you're going to get a spread of this resistant strain. So there is the emergence of a resistant strain in this.
So the resistant strain is emerging at the edges where there's the most syndrome.
Yes, and also most food.
Oh, and also, so can you tell which is important? I mean, because what we tend to be told about antibiotics, the problem is that there's too much low concentration of antibiotics, which gives the bacteria an advantage. It gives them some selective pressure without wiping them out.
So,
Whereas here is the high concentration of antibiotics, which is causing it.
well, presumably the mutant arrives somewhere else, but it can migrate now. Right? It's not guaranteed that the mutant arose over there, but that is the best situation for the bacteria to grow also. Right? If it was a mutant and I put it there, it's the best place for it to grow, as opposed to anywhere else on the chip. Right? So if there was a mutant and it somehow got there, it's best adapted to just grow and then keep going out.
So the edges are lighting up because the mutants are congregating there.
Yes, so they're outcompeting locally.
But can we tell when this mutation is happening? I mean, can we
No, no, no. Sorry?
Why is this experiment supporting use?
Right. So now this will tell you. So if I just decrease the number of initial bacteria I put, right? So there might have been some one bacteria out of a million which was resistant, and that's what's growing, right? So now I just dilute down, or rather they just dilute down to 100. There's really no mutant strain over there. You just have to wait longer, but you still get emergence of resistance. Okay? So this is the argument against any pre-existing mutation. And then, of course, this can be repeated many times. Yeah? It's just a number game. I mean, so I can literally tune down to 10, and the chance of me having a resistance in 10 is 0, pretty much. And you can do this many times, right? And you will always get this result. This I'm not so sure, but maybe you see I don't understand that, but presumably, yeah. So these are probably between half an hour to an hour, something like that. Yeah. Sorry? Uh-huh. Mm-hmm. 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 Ah. No, so the idea is that if you have a gradient, and if a mutant arose in one of them, now you have not a global competition, but a local competition, and so you create a fitter species, which can now, if it migrates to another place, can do a better job. It doesn't need for the there's no need for the mutant to arise in any one particular place. Yes. Well, I mean, it's facing the local competition in the face of this antibiotic pressure, right? Otherwise, there's no competition. If there was no antibiotic, there's no stress, it doesn't have to compete with anything. Mm-hmm. Well, okay. So if it was just one thing and it survives and it moves to another place, then it can actually do better than the other guy, right? Resistance, and so it grows. Its population grows. So wherever it grows, it has an advantage. That's about it. As long as, even if it's one, as long as there's a mutant that arises, it can do better. Yes. 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 That's what you're saying. That's what you're saying. Exactly. Okay. So what they also did was now to go take this bacteria and sequence their genomes, right? So what's being plotted here, it's a funny plot. So this is the entire genome of this bacteria, and it's basically a distance from the wild type. Okay, so you have a wild type, and you have the various mutants that arose. DG actually stands for death galaxy, is what they call the device. So they ran this death galaxy many times, and they got many of these resistant strains, all of which they sequenced, and they found that each of these strains had a mutation, a single nucleotide polymorphism in the same place, but there were 4 of them. Right? So basically, it was doing the same thing in response to the stress. It was evolving in the same way. So it was generating. So this indicates that it's somehow finding a special point where it's beneficial. And it's clearly going downhill in some landscape, right? And also they found that these SNPs corresponded to 4 different proteins. The most important or interesting of which was a gyrase, right? So this is a gyrase A. This protein is what Cipro binds to. Okay, so it has a little binding pocket for Cipro, and when Cipro binds there, it deactivates this thing, right? So these SNPs created or result in a missense in this binding pocket, which basically then prevents the Cipro from acting in the way it normally does.
How critical was choice of basically the antibiotic? I mean, what if something that can't be rectified by a single
It's possible that, I mean, this is one of the strongest antibiotics there is. And so I think it's probably the last drug you'll be prescribed. And so clearly something like this has happened with the other antibiotics already, right? Right.
Then we just won't have like a bi-state repeat that either the humans want it or humans completely run it.
You want to prevent migration, exactly. But then what this means is that you always have local niches, right? So even in your gut, it's a very heterogeneous environment already, right? Even that is sufficient for things like effects like this to happen. It's not necessarily that
Then people should stop exhorting. This whole antibiotic mafia that says don't give antibiotics. It's a lost cause.
This is what Bob says. So yeah, right. And so for him, the story doesn't quite end with bacteria. So he makes the connection actually to cancer. Because cancer it's a tumor, extremely heterogeneous environment, and then whenever you give a dose to treat this cancer, you're basically allowing biomechanisms like this for resistant cells to arise, and so by actually treating cancer, you're increasing its own survival is what he goes on with. Yeah. Well, yeah, basically he's saying that, if you give it drug, because it's this heterogeneous environment, things and cancer cells are also known to migrate. You have, no, within the tumor, right? So the chance that, you get some mutant cell to the dose that you're giving, either radiation dose or some drug you're giving, is extremely high, right? And this is happening in hours, right? And so even nothing to say that, things like this are not happening through the treatment of the patient. And right, that's the point. That's the point. Yes, that should be done, I think, and then see what happens. No, so if you give a cancer drug, if you give some drug, it kills some cancer cells, right? But it always, some cells which are resistant to the drug come up, right? And so because it's a heterogeneous environment in a similar way, right? Because, I mean, you cannot give the same dose of drug through the entire tumor. You're sending it through the bloodstream and so on. And so that creates gradients, heterogeneous environments, and so effects like this could come into play. All right. So here's another very interesting thing they found in the experiment. So when you go in and look in one of these hexagonal cells, right, they found that, so normally this is the size of a bacterium, about a micron, but they found that these cells were, a lot of long filamentous bacteria were inhabiting over there, right? So filamentation is another stress response of bacteria. So if you put it to a mild dosage of antibiotic, let's say, it somehow stops dividing. It triggers this so-called SOS response. The bacteria just keeps growing longer and longer, but it just fails to divide. Okay? So this is exactly, so in the normal condition, if you start with one bacteria, it just divides and divides and grows. But now you just put just some antibiotic, not a lot that will kill the bacteria, but some stress. They no longer divide, but they form these long filaments. Okay? But then every now and then, they will bud off a little guy. So the long filaments every now and then bud it off. Okay? And this is what they observed. Sorry? So this thing still goes exponentially. It's still exponential growth. They just don't break off into individual bacteria. It's just one long big bacterium. That's it. The mass still grows exponentially. Okay? And this is what they found, is that when you take this E. coli with its genetic material and put it with a low amount of antibiotic, it initiates this SOS response. The cells elongate, and then in these elongated cells, they saw that they have multiple copies of this chromosome. And every now and then, one of these filaments buds off a little guy, which they found is resistant to the antibiotic. And some guys just die off, right? And so eventually you get a little bacterium that's healthy and resistant, and then it starts to grow and divide normally. Okay? So their claim is now, which I find very provocative, but very interesting, is that because now it's producing multiple copies of the chromosome inside this extremely long cell, what they say is that it's producing mutant copies of the chromosome, right? And eventually it finds one which is resistant. They say that they possibly resemble a type of Turing machine where the computation of fitness is like the writing of mutations on a chromosome, and erasure of marks would correspond to the elimination of chromosomes, right? So some guys essentially die off, and recombination between the chromosomes reveals mutations. And the popping of the Turing machine upon solution of the problem would represent the picking off of the mutant, a more fit chromosome. So yes, this is now they have created multiple copies of their chromosome, because now, so normally what happens is they do create copies, but then they divide into, and they're separated. Now all of them are existing in one big guy.
The S phase is happening, the S phase keeps happening, but it doesn't never goes to mitochondria, the fusion part.
Yes. Yes, exactly. And so what they're saying is, you know, some of these guys are you know, I mean, they can eliminate via recombination the ones which are not fit and then create these mutants. That's the claim.
If you have multiple chromosomes, they all share the same cytoplasm. So what is the chance that this doesn't carry some old protein along with it?
I see. Sure, I mean, sure, you can do that. But of course, then you have diffusion, chance, all this kind of things coming in over there, right? But of course, if it's a mutant that's resistant, you don't care. Right? You just, even if it's present over there, you don't care. It's, it's happy, So what it does, what they have shown is that in response to this SOS, the bacteria overproduces these polymerases, which make copies of chromosomes. And then they're presumably making, you know, mistakes in the copies and so on. Okay? Exactly. They're trying out different solutions. Exactly. That's the claim. Okay. So now I think this is the last thing I want to say. I want to end here. I've shown you active fluids, of course, right? And the time scales are the swimming scales, are set by the swimming speeds and so on. I've also told you that you know, you can get this resistance and mutations and evolution happening on these very fast time scales given the right interesting conditions. What these bacteria also do is, you know, if you have large groups like this which are moving, it's not like just birds where they're following each other or something. They're touching each other, and those touching events lead to very interesting stuff. For example, here the outer membrane, the outside of the cell is labeled fluorescently of some cells, but not others, right? And as they touch, you see that they're passing on material. So this bacterium, which was not green, so in the bright field you'll see one of them is not green, but then the other one is, and they just touch each other, and as they touch each other, they pass on material to each other. Right? And they do this in a more interesting way, is that they actually form little tubes which connect their bodies. And they are able not only to pass membrane material, but also genetic material through these things. And as they move, they even leave genetic material behind for things, for other bacteria to get picked up and so on. Right? So this sort of goes in the direction that Chris was alluding to earlier, where, you know, evolution may be driving some of these collective or active fluid motions itself, and the other way around, where this activity can enhance some of these events and effects. Right? So this is pure speculation, but the time scales that I showed you correspond, and there's no reason why things like this cannot happen. I don't know if they do happen. Right. So this could be horizontal gene transfer coupled with things like that, where you're actually having de novo mutations.
Were these things under?
These are not. These are not. So yeah, that's what.
Since we have lunch after this, and given how interesting this was, why don't we have some questions?
They look there as if they're following trails. Do they leave Darnell-like markers behind?
They leave so-called slime trails.
Okay.
Yeah.
But then that's sort of an additional sort of thing that's different from the void-like model,
Yes.
even though it might at large scale be known.
Yes, indeed.
So these multi-nucleus filament things, is it the case that multi-nucleus cell survives if any one of the nuclei has the antibiotic resistance mutation? I mean, is it a way of kind of cooling the mutation events?
Well, presumably one of them is mutant and it buds it off. And then the remaining part, if it has something else which is mutant, also it can survive, otherwise it just dies off. But
I guess what I'm asking is does the multi-nucleus cell, you can imagine a couple of reasons. One is that if small populations are good, then sort of creating like one of the little hexagons for itself locally.
yes, yes.
But that only makes sense if the nuclei are somehow competing with each other within the cell or something like that. And then another explanation is that it only takes one of the nuclei within the cell for the whole cell survive if that one nuclei gets the good mutation, so that you are multiplying your chances of survival. So I don't know.
I don't know either, but I guess, I mean, in some sense, they are competing with each other, right? Because in some cases, the antibiotic might be doing things which prevent further processing from that chromosome, whereas for the mutant or the resistant guy, that doesn't happen. And so it's able to somehow do better, even in terms of generating division machinery and so on, right? I don't know.
Luria-Delbruck, not rule out induced mutation. So is there a scenario solution where you would actually see
So Luria-Delbruck did say that there is no induced mutation in their experiment, right? But then, in order to get or actually see this induced mutation, you need different conditions. Yes. Yes. So if you perform the same thing on a plate with a gradient of antibiotic, you'll still kill the bacteria. Somehow breaking them up into subpopulations and allowing dispersal is crucial for this thing. Right?
And they consider T1. The thing is, end of the day, the mechanism required to tackle them might be very different. Some might
Sure.
be achievable by using.
Even if you do the Luria-Delbruck thing with an antibiotic, you get the same result.
Oh.
This, of course, hasn't been done with the T1, so I don't know, but yeah.
I find completely mind-boggling that the serial writing could come up with such a standard.
So I was going to say that I find it completely mind-boggling that the Sewell Wright came up with this theory with both dispersion and gradient. So one question was, and then somebody actually proved it afterwards. That just seems completely stunning. So how did he come up with this theory? I mean, what ingredients, what was he thinking about to come up with this theory? Because it seems quite clever. And then, like, does he have a Nobel Prize, or is he going to get a Nobel Prize? Or?
No, I don't think so. But there's a history of thinking about metapopulations, MacArthur and so on, right? So, and then he brought in the concept of this fitness landscape, which is analogous to this energy landscape. And then he said that if things are playing out on this, where things are moving up and down gradients, this is what one should expect. That's how.
There was
yes, there were these metapopulation ideas. Yes, yes.
So that already there was a hint that you can get more mutation by subjecting to a...
Not more mutation, but you can get dynamics which are different. I see.
So those two experiments, one, those two experiments, one in which the first experiment where basically there is spontaneous mutation, and this experiment where showing how reduced mutation can. First, is it from the point of view of sort of dealing with the harmful bacteria, is it clear that the spontaneous mutation perhaps we can handle, but reduced mutation are more problematic from the point of view of fighting bacteria?
I think, yeah.
The second question is, if that is so, then is there a way we should make sure that the conditions that give rise to these induced mutations are not there? Because we have two situations where in one case, apparently the claim is that there is no induced mutation, and the other is that if you cleverly set up this experiment.
Well, the argument from this experiment is that it is the more realistic scenario. Whereas Luria-Delbruck is a particular case which is not, I mean, though those mechanisms do happen, that's not relevant for situations which are heterogeneous and so on, right? So the induced mutation is what we should probably expect, right? The rates and so on may somehow be cleverly tackled, and I don't know what can be done for that. But the argument is that this will happen, right? And so, yeah.
So the pheromone part is pretty interesting, like leaving the trail behind. So is there a time scale under which the pheromone will be useful for a later bacteria to pick up, or will it kind of get diffused and be useless? So the concentration is not high enough.
Right.
No matter what pheromone trail you leave, it will not be useful for later.
Right. So what these guys do is they actually leave a slime trail behind, meaning they're actually leaving a lot of material. Yeah, no. So they leave a lot of material behind, including genetic material, membrane material, all sorts of things. Okay, and clearly there is going to be a time scale associated with the degradation of those materials and so on. But then they encounter their trails quite frequently.
So the mobility time scale and the pheromone relevance time scale are kind of compatible. One is not too high.
But it's also not clear what those things are doing. We only know that they're being left behind, and some hints of the kinds of things being left behind. But what they're doing, what role it's not known. No, I don't.
We can say that how the probability of this disease is due. So we are supplying antibiotics. Antibiotics supplied there.
For the cancer, you mean?
Cancer.
So there's a cancer drug.
There is a case that we are even cases with
Yes, that's the argument.
the chance of that disease we are supplying.
I do.
Statistical approach, that statistically we can say
I don't know, and I don't. I don't think they have any data from real tumors, first of all. But they've just started doing some experiments with cancer cells in these environments, and they find similar things happening where mutants arise and so on. But the mechanisms presumably are very different. And I don't exactly know what you mean by statistics of the disease being cured. Yes, yeah. I think I don't think I get your question. Sorry.